Hello students, welcome to this tutorial. So basically we are going to focus on the contraction of skeletal muscle. So we want to combine the excitation and then how does excitation brings about contraction. So this is excitation, contraction, coupling. This is basically the contraction of skeletal muscles. So now with the excitation, contraction, coupling, there are some basic information that you should know before even start listening to this tutorial. So the first thing is you must be able to familiarize yourself with the neuromuscular junction and how electrical impulse is able to move from one neuron to another. Now we are talking about a special synapse, which is a neuromuscular junction, where um, the transmission of electrical impulse need to move from the neuron and go into the muscle so that the muscle can be depolarized and bring about what? Contraction. I'm going to explain this step by step, but hang on just a bit. So now, the first thing is synaptic transmission. You should be able to understand it. Then from there, you should be able to understand the structure of the skeletal muscles. Moving from the epimysium, perimysium, endomysium until we reach the myofibril, and then now we go reach the specifically the myofibril is in the muscle fiber. So now this myofibril, that it contains filaments, okay? And specifically we said the structural and the functional unit of skeletal muscle is what we call the sarcomere. The sarcomere is made up of two types of filaments. That's the thin filament and the thick filament. These two are what really facilitates in the contraction of a skeletal muscle. If you have that background knowledge, then we're good to go. Let's start now. So now, we should be able to know that the process of excitation, contraction, coupling begins with the release of acetylcholine from the motor neuron into the synaptic cleft. The release, you are not going to explain this as if you're explaining the electrical transmission. You are going to start from the release of acetylcholine into the synaptic cleft. Okay, so acetylcholine is going to be released in the synaptic cleft. So now let's start with one. Release of acetylcholine into the synaptic cleft. So what happens? The motor neuron is going to release acetylcholine into the synaptic cleft through exocytosis. We know this one. We are talking about the neuromuscular junction where the neuron and the muscle meets. Then we know that acetylcholine is a neurotransmitter that plays a crucial role in transmitting signals from neurons to neuron. This summary is how you are going to write whenever they tell you to write the process of excitation, contraction, coupling. If they tell you to write it with the aid of a diagram, you are just going to draw the neuromuscular junction and label important parts like the channel, the presynaptic neuron, and then you are going to also label the muscle fiber specifically here, the muscle fiber. Okay. So on the release of acetylcholine, you should be writing it like this. Release of acetylcholine. Then you start explaining under release of acetylcholine. What happens? The motor neuron releases acetylcholine into the synaptic cleft through exocytosis. So acetylcholine is a neurotransmitter that plays a crucial role in transmitting signals from neurons to muscles. Then you go to 2. What happens? Once this acetylcholine, remember, let me visualize this thing. This is we are talking about the neuromuscular junction. So your mind should be here. Your mind should be here. Okay. The blue one is the postsynaptic cell. Probably in this case, we're talking about the muscle fiber. The red one is a neuron. Now they are meeting here. Acetylcholine, once it is in the synaptic cleft, what happens? Acetylcholine, you are going to write it binding of acetylcholine to receptors. So acetylcholine is going to bind to nicotinic receptors on the surface of the muscle fiber. You get it, right? This is the muscle fiber here. So it's going to bind to the nicotinic receptors on the surface of the muscle fiber. This binding causes a conformational change in the receptor. What is this conformational change? We are talking about the opening of its ion channel. Specifically, here we are talking about the opening of what we call sodium voltage gated channels. Then we have this nicotinic acetylcholine receptor ion channel are now open. These are basically the sodium voltage gated channels in the muscle fiber. Once they open, then we go to the next step, which is the depolarization of the muscle fiber. You know, when the channel is open, probably there's going to be sodium rushing inside the cell. So sodium is going to rush inside the muscle fiber, and inside is going to become more positive. So you need to use physiological term when you're explaining here. Once the channel opens, sodium rapidly rushes inside the cell, making the cell more positive. More positivity, that's depolarization. So depolarization of the muscle fiber. So what happens in the depolarization of muscle fiber? We now want to explain. The opening of nicotinic acetylcholine receptor ion channel allows, these channels we're talking about is a sodium channel. Let's not get confused. These are sodium voltage gated channel. So once they open, they allow an influx. Influx means inside movement of ions, meaning ions are moving from outside to inside. So this allows an influx of sodium ions into the muscle fiber. This influx of positively charged ion depolarizes the muscle fiber, making it less negative or making it more positive. We are talking about one and the same thing, because we know that inside is usually negative, outside is usually positive. So when sodium rushes inside the muscle fiber, what happens is that inside it becomes more negative, or rather more positive. I mean, less negative, or rather more positive. Then after that positivity, you know, when ions have accumulated, like sodium was accumulated, what follows is an action potential. So we are going to indicate generation of an action potential. If the depolarization is strong enough, there's a condition. If the depolarization is strong enough, the depolarization we're talking about is the influx of sodium inside the muscle fiber. If it is enough to produce an action potential, then if the depolarization is strong enough, it triggers an action potential in the muscle fiber. Okay? Muscle fiber and muscle cell, we're talking about one and the same thing. The action potential is a rapid change in the membrane potential that travels along the length of the muscle fiber. Along the length of the muscle fiber. Remember, the structure is like this, right? We have T tubules here. So the first step is that whenever an action potential has been generated, what it travels along, okay, along, before it can use the T tubules to move downwards, it first travels along the length of the muscle fiber. I believe we are together there. Okay, so we have generated an action potential because the stimulus is enough to generate an action potential. It's rapid, like. So now there's going to be a change in the membrane potential. That's what we describe to say is an action potential, because there is an equal distribution of ions, and that brings about potential difference. Okay, get what I've written here: generation of an action potential. Summary: if depolarization is strong enough, it triggers an action potential in the muscle fiber. The action potential is just a rapid change in the membrane potential that travels along the length of the muscle fiber. Then let's now go to release of calcium ions. Okay, so now understand here. Where have we reached? These are bundles of the muscle cell, and we have the T tubules in them. They are transverse tube because they move perpendicular to these longitudinal line of the muscle fibers. So what an action potential at this particular time has done is it has just moved along the length. It is just traveling along the length of the muscle fiber. It has not moved downwards yet. So what happens during the release of calcium ion? Let's read what happens here. The action potential reaches the T-tube. The action potential which is coming here, it reaches this T-tube now. Okay, it's coming. It reaches the T-tube, which are specialized structure that penetrate deep into the muscle fiber. As you can see here, they penetrate deep into the muscle fiber. Okay, so it's going to move down the T-tube towards the muscle fiber, or penetrating into the muscle fiber using what? Or moving, it's going to move along the muscle fiber and downwards the T-tubes. So in these T-tubes, I said they are what we call dihydropyrimidine receptors. And where are they located? The dihydropyrimidine receptors are located along the walls of T-tube. These receptors, it's very much important that you know it, that they are voltage regulated, meaning they respond to a certain voltage. So if that action potential travels, if it travels along the muscle fiber and down the T-tubules, what happens? If an action potential has reached the T-tubules and this dihydropyrimidine receptor is voltage regulated, meaning it responds due to change in the potential, due to potential difference happening due to ion distribution, inside becoming less negative or more positive, and outside becoming more negative or less positive. So now, since these receptors are only regulated by voltage, this means they respond to change in voltage due to action potential. When action potential comes, they open. You get it? When an action potential comes, they open. Now, one other important fact here when we're talking about this point is this. We are on the release of calcium ion. So bear with me, we are still here. One important fact about dihydropyrimidine receptors is this. They are mechanically connected with what we call the ryanodine receptors. If you haven't mentioned dihydropyrimidine receptors and ryanodine receptors in your explanation for muscle contraction, then we don't know where you got that kind of an explanation. Because these receptors are very much important. They are the ones which control whether calcium is going to remain in the sarcoplasm, okay, or it's going to be released. So the dihydropyrimidine receptors respond to voltage, but it is mechanically connected with the ryanodine receptor. When we say mechanically connected, we mean to say the ryanodine receptors are moved manually by dihydropyrimidine receptor. They don't use chemicals like synapse or electrical, no. Okay, it's more like you connect two doors of the house, and you opening the first door is you have put a wood in between. Immediately you open the first door, you push it, is going to push another door. That's the connection that is there between the dihydropyrimidine receptors and what we call the ryanodine receptors. I hope we are following. Now the action potential has moved down now, right? And it has come in contact with the what? With the ryanodine receptors, making them. What happens? Let's see what happens. Okay? We are still on release of calcium, and this is point number 5. So once the dihydropyrimidine receptors open due to that action potential, due to the change in voltage rather, it mechanically opens the ryanodine receptors on the terminal cisterna of the sarcoplasmic reticulum. Let's get this clear. They are going to ask you about the location where the dihydropyrimidine receptors are found. Please answer this way. The dihydropyrimidine receptors are located along the walls of the T-tube. This is the reason why it easily responds to change in voltage. Change in voltage, we're talking about action potential. And it being mechanically connected with the ryanodine receptors on the terminal cisterna. The ryanodine receptors, they are on the terminal cisterna. Let me draw it. This is sarcoplasmic reticulum, and these are projections. These projections are what we call terminal cisterna. And exactly here is where the ryanodine receptor is located. And then the T-tube is here. Then we have what we call the diidopyrimidine receptor is here. And then what happens is that they are mechanically connected. This automatically opens the ryanodine receptors. You remember the sarcotubular system? What did I say is a sarcotubular system? That is a triad. Let me draw the triad once again so that we're moving together. The triad is like this. We have the T-tube, okay? And then we have the two sarcoplasmic reticulum, okay? With these projections. These projections, we are going to call them terminal cisterna. These are terminal cisterna. So when they ask you, where are the ryanodine receptors? The ryanodine receptors are found on the terminal cisterna of the sarcoplasmic reticulum. This is a sarcoplasmic reticulum. In other words, it's a cytoplasm. So now, since this is a T-tube, this is a sarcoplasmic reticulum, and the diidopyrimidine receptors are along the T-tube. They are mechanically connected with the ryanodine receptors. Diidopyrimidine receptors mechanically connected with the ryanodine receptors. So immediately this receptor open, it automatically opens this receptor. Why? What are the sarcoplasmic reticulum? Sarcoplasmic reticulum stores calcium, which is very much important in muscle contraction. Let's see how important is calcium in the muscle contraction. I believe now we are together here. The sarcotubular system, this is a triad where two sarcoplasmic reticulum meet with the T-tube. Hence a triad. Tri means 3. So now, once the diidopyrimidine receptors open, due to change in voltage, it mechanically opens the ryanodine receptors on the terminal system of the sarcoplasmic reticulum. This allows calcium. Okay? Let me draw it again. Okay? Calcium is here. Calcium is here. Then the ryanodine receptor is here. Immediately it opens, it allows calcium to flow out of the sarcoplasmic reticulum and go outside, which is the cytosol. Okay? I believe we are together now. So, this allows calcium ions to rush into the sarcoplasm, which is the cytoplasm of the muscle cell. The sarcoplasm, which is the environment, outside environment, from the sarcoplasmic reticulum, going into the cytoplasm of the muscle cell. If they say sarcoplasm, just know we're talking about the muscle cell. I mean the cytoplasm of the muscle cell. So now, this allows calcium ions to rush into the sarcoplasm. Let's see exactly. Let me explain a bit here. This is an action potential that was generated, remember, due to acetylcholine which came. It moved along the muscle, the muscle fiber and down the T-tube, causing the diidopyrimidine receptors to open, mechanically opening the ryanodine receptors, which are on the terminal system of the sarcoplasmic reticulum, releasing calcium. Okay? Releasing calcium into the sarcoplasm. Sarcoplasm is the cytoplasm of the muscle cell. We should familiarize ourselves with that. So now, what is in the sarcoplasm? Why should calcium go into the sarcoplasm? That's where now our area of interest is coming. In the sarcoplasm, there is what we call troponin. Before I say anything about troponin, remember, what is troponin? What is troponin? What is troponin? Troponin is part of it's a thin filament. It's a thin filament. We said parts of the thin filaments is made up of the troponin, tropomyosin, and actin. Actin is the active site of where the thick filament bind, which is specifically the myosin. Thick filament is made up of amyline, it is made up of titins, and it is made up of myosin. We should never forget such things. Questions will always arise. Okay? So troponin basically in the sarcoplasm is of three forms. We can see troponin I, troponin C, and troponin T. Troponin T is tropomyosin. I'm going to explain this. But here, let's focus our mind on troponin C. Troponin C. What the hell is troponin C doing here? Troponin C. Troponin C is calcium binding. Calcium need to bind to troponin C. What happens? What happens when calcium binds to troponin C? I know you want to know what happens when calcium binds to troponin C. Let's see what happens to calcium. What happens to troponin C once calcium has bound to it? So let's 6.6. Binding of calcium ions to troponin and tropomyosin. Let's see what the effect. The released calcium from the sarcoplasmic reticulum via the ryanodine receptors, and now calcium is in the sarcoplasm, also known as the cytoplasm of the cell of the muscle cell. So the released calcium ions bind to troponin C and tropomyosin, which is troponin T, causing a conformational change. It causes a conformational change. Immediately calcium binds with troponin C specifically. This causes a conformational change. What is the conformational change? What kind of an animal is a conformational change? Is it an A+? Is it a B+? No. The conformational change, this conformational change shifts the position of tropomyosin, exposing the myosin binding site on the actin filament. What the hell? This doesn't make sense. So now the thing is this. Tropomyosin at rest. Let me read again so that we go to the next slide. You see what happens with tropomyosin. The released calcium ion bind to troponin C and tropomyosin, causing a conformational change. This conformational change shifts the position of tropomyosin, exposing the myosin binding sites on the actin filament. Okay, let's see what happens. Binding of calcium to troponin C. What happens? Let's see the first image. In the first image, we said actin and myosin, they should bind. But on the active site here, you can see this purple thing. I'm not very good with colors, but yeah, something like that. Purple here. This is where the actin, this is the active site for actin, where tropomyosin should bind. But at rest, tropomyosin is the one which is bound to it. Tropomyosin is the contractile protein that covers the active site of actin when the muscles are relaxed. You're going to find such questions in MCQs. What covers the active site of actin of the skeletal muscles at rest? Then they put troponin. It's tropomyosin. So now, what happens when calcium binds to it? Upper, it has not bound. There is no binding here. We'll see what happens when calcium finally binds to it. This is just a demonstration. What happens when calcium binds to troponin C specifically? What does it do? what does it do? It causes a conformational change. What is this conformational change? The conformational change that we're talking about is that the tropomyosin moves out of the active site of actin. Actin is a thin filament, myosin is a thick filament. I'm going to repeat this because questions are going to come. So if this myosin and actin are bound, they are bind, then the effect is greater that it can even now use energy to move those Z lines towards the center, to move sarcomere, one Z line, from one Z line to another Z line, that's a sarcomere. When we want muscle to contract, we need to shorten the size of the sarcomere. Its length, we need to reduce it. That's what we call muscle contraction. So we're going to look at it how exact. Calcium was bound to troponin C in the sarcoplasm, and it has caused the conformational change. Tropomyosin moving out of the active site of actin filament. Wow, this is nice. Calcium is really important. So now, what happens? What happens when myosin and actin are bound? They are in contact. Whenever myosin and actin are in contact, it brings about muscle contraction. So now let's read this. Muscle contraction, the exposed myosin binding site, which is the myosin binding site, we mean the actin active site for myosin. Actin is the one which is having active site, has now been exposed due to calcium binding on troponin, and that conformational change has occurred. We've seen tropomyosin moving out of the active sites of actin filament to allow myosin to bind to it. So the exposed myosin binding site allow the muscle filaments, actin and myosin, to interact, leading to muscle contraction. It's simple. It's so simple. You've gotten it, right? Like very, very simple. The coordinated movement of the filament is going to be driven by what we call myosin heads, which use energy from ATP. Okay? So now, I want to mention something here. This that they are formed, we call it cross bridge. Okay? The cross bridge is this complex when actin and myosin, they are bound together. But the major component of the cross bridge is myosin. We should take note of that. The major component of this contact is myosin. So the coordinated movement of filament is driven by the myosin heads, which use energy from ATP hydrolysis. ATP hydrolysis, we mean to say we break it down, because ATP need to be hydrolyzed into a phosphate and what we call adenosine diphosphate. A diphosphate. After this, what happens? So now let's get this. ATP binding reduces affinity for myosin, actin binding. ATP is converted into ADP and a phosphate by what we call ATPase enzyme. ATPase enzyme. Okay? Then now muscle contraction. So now on we continue now. We want to look at how does energy get to be provided. You need to explain this well. It's simple. Okay? What we are saying is ATP is going to be hydrolyzed, meaning we're going to break it down. We have phosphate. Let me just write abbreviations. We have phosphate and we have adenosine diphosphate. Di means 2 plus a phosphate. That's what makes up the what? The ATP. So ATP is going to be hydrolyzed so that it can be used. So all these are important. We now have a phosphate and we have ATP, right? These had been hydrolyzed. So let's see what happens when ATP binds and when phosphate binds. We want to understand that. Now let's get these two. 1, 2. When ATP binds and when phosphate binds, let's see what happens. ATP binding reduces affinity for myosin actin binding. Meaning, when this ATP binds to the myosin head, understand me clearly, what happens is it more like want to create that detachment between the myosin and actin. This is what we mean. ATP is converted into ADP and phosphate by ATPase enzyme. Once phosphate detaches from ADP, the muscle heads pull the actin. This is called the power stroke. What is a power stroke? Power stroke is when phosphate has detached itself from the myosin head. If it detaches itself from the myosin head, then the myosin causes a move. Like this detachment causes a move. Myosin moving to the left or right. What we want is towards the M line. We want the myosin on the right to move towards the left, and we want the myosin on the left to move toward the right. Moving the muscle, the sarcomere, towards the M line. Okay? From the binding of myosin and actin up to the power stroke, the process is called the sliding filament. Let me demonstrate this. Let me find a perfect place where to demonstrate this. Okay, let's do this. We have the sarcomere here. Right? This is a Z line and this is a Z line. And we have M line here. And then within these M lines, we have these major components which are having the what we call myosin head. Right? Myosin is in this area. And then let me use this to draw actin. Right? Actin is in these areas. Actin is in these ranges. This is to ease. This is the active side so that they can easily bind with the. So we're going to get this area. This is myosin in red. The actin is in blue. So want them to bind. This is what is going to happen here. This is what is going to happen here. Even here. Even here. Even here. So what we want to do is we want the movement. We want the myosin heads to move towards the left when it's towards the right, and here to move towards the left. Then they are moving towards the M line. Okay, they are moving towards the M line, and this is what brings about muscle contraction. Meaning we are going to shorten the sarcomere. It's going to be somewhere here because we are moving these strengths here because it's flexible. Remember those 4 properties of muscle? It's flexible and it can be stretched. So it's going to move towards, and this is going to bring about muscle contraction. So the sliding filament, when we say the sliding filament is that, let me draw it now here. If this is the myosin, sorry, not the myosin head. This cannot be myosin head. If this is the active site, if this is the active site for actin, and then let me draw it this way. And then we have this is myosin and it is having this head. You understand, eh? It's having this head. This is connected here. This is actin. This is myosin. Okay? So we said what happens when ATP binds? When ATP binds, what happens is that myosin loses affinity. This is myosin. It loses affinity with actin. So ATP brings about this problem. Then let's see what happens when ADP binds. When ADP binds, it increases the affinity between actin and myosin. But remember, this ADP is with a phosphate from the hydrolysis of ATP. So when ADP with phosphate combined, they bind to myosin, the affinity increases. But what happens is this: when a phosphate detaches from ADP, what happens is this: it causes the myosin head to move towards the right, towards the left here. It causes the myosin heads to move in this direction. This is what we call power stroke. We want to move this actin, we want to move this string. If it was here, then it would look like this. If this myosin moves towards the left, it's going to look like this. It's going to look like this. Since it's here and the myosin is here, now it's going to look like this. This is going to be the myosin head, and this is going to be the active side, like active side. You've seen the angle? They have moved from here, they have moved to here, meaning we have moved this towards this end. You understand? So this is what we call power stroke. And power stroke happens when phosphate group detaches from ADP. When phosphate detaches, then there is a power stroke. And then what happens is this. What is the sliding filament? The sliding filament that happens is that myosin and actin slide past each other. They slide past each other, then making them like this on top here. Then we come now to what we call walk along theory. Walk along theory. What is walk along theory? For muscles to continue, it means the myosin is going to be moving here and another myosin is moving here in a continuous manner. So now imagine myosin is moving, this myosin is moving towards the left, then the other myosin is moving actin towards the left, then it happens on the other side here, it moves towards the right. Then it's more like they are moving from right to left, and there are a lot of them bound. It's more like they are walking. That's what we call walk along theory. Okay, so this is how. So now let me finish. Once phosphate detaches from ADP, the myosin heads pulls the actin. It pulls it. If it was from the left, then it's moving it towards the right. The one which is at the end of the Z line at the right, the myosin is going to move that actin towards the left. We want to these things. We are moving everything towards the M line. So the myosin heads pulls the actin. This is called the power stroke. From binding of myosin and actin up to the power stroke, the process is called the sliding filament theory. They are moving past each other. They move past each other. Then, however, the phase from one power stroke to another is called walk along theory. Meaning, it's more like it's done now in a more continuous manner. The myosin is moving the actin towards the left or towards the right. It depends with where this myosin is. But the aim is so that we can move these things towards the M line. This is what we call walk along theory. Therefore, ATP hydrolysis produce force and shorten the muscle fiber, bringing about muscle contraction. How does it shorten? Let's pay attention to how it shortens. How it shortens is this. Remember, this is the M line, this is the Z line. And maybe probably it was here. The Z line, the initial Z line was here. But because myosin moved actin towards the left and here it moves actin towards the right, then they shortened the muscle. You can see these arrows. They have shortened the muscle. From this sarcomere, now we have a subsarcomere which is looking like this. We have shortened the muscle. When we say walk along theory, we mean to say actin has bound to this and it is moving towards this direction. It moving towards this direction, it's more like walk along theory. Cross bridge is this combination. Actin and myosin binds. ATP, when it binds, it reduces affinity. You've seen, it has detached it. Then this is not good. We need to hydrolyze ATP to phosphate group and adenosine diphosphate. Then once they bind to ATP, they increases the affinity. ATP moves towards the actin. Then the problem here is that phosphate knows that if at all I'm still attached to ADP, I'm just making the affinity to be stronger. So what I should do is I should move away from ADP so that the myosin can move the actin towards the left. As you can see here, look at here, it has moved. This is what we call power stroke. It was this simplified way. Thank you very much for watching. And if you are still watching this video, probably it means you have benefited a lot. If you haven't subscribed, make sure that you subscribe to the channel. Support us and continue making more tutorials this particular way.